Right, well, good afternoon, everyone, and welcome to this very special edition of Bite-Sized Corrosion. My name is Vanessa Seeley-Fisher, and it's a real pleasure to be able to host today's session. In every session of Bite-Sized Corrosion, we try to break down another aspect of corrosion-related information into what we hope are interesting and informative and, shall we say, palatable nuggets, in the hope that you will leave us having learned something new and eager to continue your journey in understanding corrosion just that little bit more, and hopefully making your corrosion experience kind of easier to digest. Now, I am absolutely delighted today to welcome Dr. Reza Javaherdashti, and I would say he's from all over the world, but currently he's live in Croatia, and he's going to be spending time today talking to us about cathodic protection and microbial corrosion. And it's a real delight that he has chosen to spend this time with us. Reza.
Thank you, Vanessa. Good afternoon, everyone. I'm so glad that I had the opportunity of coming to South Africa again, this time virtually. And I hope that in the future, it will be actually a real meeting between us. Without wasting too much time, I start presentation. All right, so we start with this word of wisdom from Benjamin Franklin that is actually putting emphasis on actually practical aspects of what we learn. And I hope that what we will discuss today will also give you some idea for finding solutions to your day-to-day responsibilities. The presentation today will have actually 8 main sections. We will start with the challenge that I'm sure you have all received, and some of you have tried to actually solve it. We will discuss it at the end. After a brief self-introduction, we will delve into the more technical issues related to this presentation. The topic, as you know, is cathodic protection and microbial corrosion, a never-ending challenge. And here we will actually focus on a very important aspect of an issue, a corrosion issue, that I would say is not that known. Actually, during my professional life, I have met at least 4 types of corrosion engineers. The first type, they totally deny that bacteria are capable of inducing or affecting corrosion in any way. Then comes another group who know that, who appreciate that bacteria are actually capable of doing something, but all they know is just SRB, sulfur-reducing bacteria. A much higher level of understanding about MIC or microbial corrosion is that in addition to SRB, there are others like iron-reducing bacteria, sulfur-oxidizing bacteria. And to me, the highest level will belong to those who also appreciate the role that bacteria such as Clostridia can play in microbial corrosion. I will go through some of bacteria that may be very interesting from microbial corrosion point of view, and then we will talk about cathodic protection and other stuff. Bear in mind that a corrosion specialist with MIC expertise need to be aware of non-MIC corrosion processes. So if I am a corrosion specialist, but not aware of, say, corrosion under insulation, or atmospheric corrosion, or under deposit corrosion, and so on, then I will not be able to understand microbial corrosion well. But the opposite is not necessarily true. Just because someone knows non-MIC corrosion does not necessarily mean he or she knows MIC. A brief self-introduction. As I said, my name is Reza, surname is Javaherdashti. I have a PhD in corrosion from Monash University in 2005 in Australia, and I did my postdoc at Curtin University, again in Australia in 2010. I have been involved in more than 400 corrosion management projects worldwide since 1993. I am approved by the US ASME and also Society of Petroleum Engineers as a corrosion, particularly MIC lecturer. So far, I have had more than 5,000 hours of quite successful training courses about corrosion management, electrochemical corrosion, and microbial corrosion. I have authored several research papers, root cause analysis reports, and books. At the moment, I am doing consulting work on corrosion in general and microbial corrosion in particular for many reputable companies around the world, from Australia to Malaysia. These are some of my books published by reputable publishers, such as Elsevier, CRC Press, and so on. And from time to time, when it is necessary in my slides, I will quote my books. Let's start with definition of microbial corrosion. Microbiologically influenced corrosion, as NACE, now AMPP, addressed it in 1993, is a type of corrosion that can be initiated, enhanced, or even inhibited by, but not limited to, bacteria. So in addition to bacteria, you could also have the action of archaea, lichen, algae, fungi. So it is not just bacteria. But during this presentation, we will only focus on bacteria. Almost all engineering materials are vulnerable to microbial corrosion, both metals and non-metals, such as concrete, even glass, polymers, composites, almost all types of steels, stainless steels, they are susceptible to microbial corrosion. The cost of microbial corrosion is normally around 20% of the cost of corrosion. However, technically speaking, the cost of microbial corrosion could be even around 40% of the cost of corrosion. In order to have a rough idea about how big this figure is, take the GDP of South Africa and just assume that the cost of corrosion is just 5% of it. This is an assumption. The real figure could be much higher, okay? When you calculated that, the figure you get will be an estimated figure of the direct cost of corrosion to South African industries. Then just take 20% of it, although the real figure could be much higher. And this can give you an idea how much microbial corrosion is actually doing harm to your industries. In these paragraphs, 4 paragraphs that you read here, I deliberately made a mistake. This is something that from time to time I do in my presentations, so that this will be a challenge for the audience. They will not think that they have learned everything they needed from the presentation. I'm trying to give them some sort of challenge. So what do you think could be wrong, or the mistake I deliberately made here? In order to understand that, I recommend you to read my book, Corrosion and Materials in Oil and Gas Industries, published by CRC Press. There I have an entire chapter about microbial corrosion, and I have also explained why there is a mistake here. A few case histories before we start. These case histories have been taken from my book, Pathological Mini Atlas of Microbiologically Influenced Corrosion and Deterioration, MIC and MIT. In that book, I enjoyed collaboration from many of colleagues from all around the world who kindly shared some of their case histories with me. And here I'm sharing with you just two of them. One case is about this steel sheet pile from a cave wall. The time in service has been more than 20 years. The environment has been marine environment. The cause of failure has been the activity of sulfur-reducing bacteria, SRB, APB, acid-producing bacteria, IRB, iron-reducing bacteria, SOB, sulfur-oxidizing bacteria, and IOB, iron-oxidizing bacteria. We do have a lot of these bacteria in marine environments, as we will see in a moment. The treatment has been a replacement of the part, also application of cathodic protection. Then we have this picture from steel H pile, bridge substructure. The material has been carbon steel. It has been in service for 45 years. And as you could see, it experienced severe pitting. Environment has been estuarial brackish water with a salinity of around 30 grams per liter, which is quite high. The cause has been, obviously, causes has been microbiological influence corrosion, facilitated by marine fouling or biofouling. And the treatment has been by installing cathodic protection. This is an example of biofouling or macro marine fouling on strainers on an offshore topside. And this is a very important issue for especially offshore structures. And I will also try to describe it briefly. Before we start our presentation about microbial corrosion, we have three brief technical introductions that we need to cover. One is the approaches towards microbial corrosion. There could be two approaches. One approach focuses on the material, another approach on the system. The material-based approach is interested in understanding what a particular material could do about or its behavior against microbial corrosion. For example, I may say that I'm interested in knowing how carbon steel can do against, for example, corrosive action of sulfur-reducing bacteria, or stainless steel 316L. So this is a material approach, and most of the time, this is an academic approach. Researchers, academia like this type of approach. They take a material, certain material, they take a certain group of bacteria, and then they try to find out how that material actually behaves. A system approach is that instead of the material, you think that, okay, it is the system I'm interested in. For example, the pipeline I'm responsible for, the cooling system, the steel piles of a jetty, for example. The main issue here is that if I know under what conditions, for example, carbon steel can become susceptible to microbial corrosion, it really doesn't matter in which system it's being used, either as a pipeline or cooling system, heat exchanger. As long as I know the issues that could interrupt with the performance of the material, then the rest is easy. Working conditions only dictate the constraints where the material can act. This is fact number one. An example of material is, for example, take stainless steel. Most of people, when they hear stainless steel, they think that they never corrode, which is quite wrong. We have 5 groups of stainless steels, like ferrites, ferritic stainless steel, austenitic stainless steel, and based on iron-carbon phase diagram, you can see that we could recognize austenitic stainless steel and ferritic stainless steel, as shown by these two arrows, red arrow and black arrow. One group of stainless steels is the so-called martensitic stainless steels, martensitic steels. But on this diagram, you don't see martensite. You do see austenite, you do see ferrite, but you don't see anywhere martensite. My question to you is, why martensite is not here?
It's not an equilibrium phase, and this is an equilibrium diagram.
Perfect, because martensite is a non-equilibrium phase. And what we just saw was an equilibrium phase. Thank you. Okay, that was an example of material. Now let's go to industry. For example, offshore production industry. The red arrows that you see and the red circles actually designate where you can expect microbial corrosion to happen. So, for example, for offshore structures such as platforms, you could expect microbial corrosion to happen on both top sides and the submerged section. For the FPSOs, pipelines could be susceptible hot spots. When it comes to onshore industries, especially upstream, you can see that where these arrows are shown, where microbial corrosion can be a problem. So based on material and industry, you can think about possibility of microbial corrosion. There is a difference between likelihood and risk. What it is, you all know this risk assessment matrix, where the likelihood of a danger is tabulated against the consequences it could have. And risk is defined as a byproduct of the likelihood and consequence. Likelihood of microbial corrosion is not the same as its risk. You may say that the likelihood of microbial corrosion in my system is unlikely because I'm using cathodic protection, because I'm using biocides or coating. Even if the likelihood is unlikely, because of the consequences it could have, the risk could always be high. So risk and likelihood could be quite different from each other. Just because likelihood of MIC in your system seems to be unlikely does not necessarily mean that its risk is high. The consequences could be of economic, ecologic, or even safety type. It is always a challenge between corrosion effects, corrosion impacts, environmental aspect, and environmental impact that I have discussed in my book, A Global Warning on Corrosion and Environment, that in its category was first, and it was published in 2010. For the first time, we addressed the relationship between corrosion, mutual relationship between corrosion and environment. Then there is this difference between series and parallel corrosion reactions. In no book you can find such a distinction. And I have explained this in my book, Corrosion Policy Decision Making, published by Wiley. Corrosion reactions could be divided into two geometries, let's say, series and parallel. When we say series corrosion reactions, it means that you have at least one culprit, one main corrosion process, and it actually, and other corrosion reactions are being resulted from that main culprit. So if you can locate that one, the main corrosion reaction, and treat it, the whole corrosion problem in your system will be solved. Contrary to that is parallel corrosion pattern, where you could have more than one corrosion reactions taking place independently from each other. And even if you happen to solve one, does not necessarily mean that you have solved the whole problem. An example is, for example, a buried pipeline, all right? You could have external corrosion and internal corrosion. They are happening in parallel. Just because you have cathodic protection on external surface of the pipe, it doesn't have anything to do with internal corrosion problems, and vice versa. At the same time, what you have in the pipe could be taken as an example of series corrosion reactions. Like, for example, you have done hydrostatic testing on the pipe, but for some reason or another, some water is left inside and a biofilm has been developed. The biofilm actually can be leading into microbial corrosion issues, and these microbial species could actually focus on heat affected zone, on weld zone, and cause leakage. So you have a chain of events. If in the first place you can address the biofilm issue, then chances are you wouldn't get post-hydrostatic testing microbial corrosion. There are a lot of examples as such. By the way, for the challenge we mentioned, please note the difference between soil particles in diameter in sand and in clay. This is a very critical issue when we talk about the solution to our challenge. Then some quick microbiology. I'm not going through all these microbiology because I know that you have heard a lot about this. What I'm trying to do is just to direct your attention towards engineering importance of it. We can have cells like human cell, animal cells, plant cells, for example, that are referred to as eukaryotes. And then we have prokaryotes, such as bacteria and their cousins, archaea. Now, why I say archaea and bacteria are cousins? Because they are just like left hand and right hand. They are similar, but not the same. So there are many cases, like, for example, high temperature, where archaea could be active, but bacteria cannot, and other issues as well. There is also one important issue here, that no microbiologist, even those who call themselves MIC experts, have ever addressed it in their notes, simply because they don't know it themselves. It is horizontal gene transfer. What does it mean? In simple engineering terms, it means this: a group of bacteria that we think they can do just a specific work, under certain conditions are capable of doing other bacteria work too. Clostridia are anaerobic. Anaerobic means they don't like oxygen. Are anaerobic spore-forming bacteria that can produce organic acid and also produce ample amount of hydrogen. Therefore, they can cause hydrogen-induced cracking. However, under certain conditions, Clostridia can do iron reduction. Although iron reducers are aerobic, they like oxygen. Be aware of this term, horizontal gene transfer. If you think I know SRB. No, my friend, you don't know, because these guys have been around for billions of years. And during these years, they have developed very sophisticated defense mechanisms that we have just started to learn. Factors by which microbiologists classify bacteria are these 7 factors. I don't have time to go through all of them. However, I will just explain 2 of them, which also have engineering importance, oxygen requirement. Some of these bacteria are called obligate aerobes. They require oxygen, obviously. Some of them are microaerophils. The name implies micro, small, aero, air, fills, loving. They require a little bit of oxygen. Then obligate anaerobes. They don't like oxygen. Then the most dangerous ones, facultative anaerobes. They can switch between anaerobic and aerobic. And capnophils that require CO2. Why I'm putting it here? Why I'm putting these guys here, capnophils? In industry, when they do hydrostatic testing, and you know what it is, they fill a pipe, for example, with water, and they increase the pressure to a certain level, normally 1.1 times of maximum operating temperature, just to find out the strength of the weld and if there is a leakage. And these two makes it different from pneumatic test. Anyway, when they dry and drain water from the pipe, one of the practices is to fill the pipe with a neutral gas like CO2. It is funny, but yes, in their standards, our beloved mechanical engineers, especially, they call CO2 a neutral gas. However, anyone who has taken an elementary course in chemistry knows that CO2 is not neutral. Argon is a neutral gas. However, the issue is not this. If for some reason you have left some water, some water ponds inside the pipe, in which chances are you have capnophils, then by pumping CO2 into the pipe, you are actually feeding them. Then what happens when the pipe goes into the service, or sometimes even before going to the service, it develops a lot of leakages. Metabolism. Now, one issue with metabolism is that bacteria could have aerobic metabolism like us, human beings. We use oxygen in order to burn food and produce energy. But when it is anaerobic metabolism could be divided into anaerobic respiration, that sounds like an oxymoron, respiration, anaerobic, and anaerobic fermentation. I'm not going through fermentation. What is important is anaerobic respiration. In the same way that in electrochemistry, we have anode and cathode. Anode is the electrode from which electrons are sent off. And cathode is the electrode where they are received. We have anode and cathode in biological systems too. They are called as electron donors and electron acceptors. Electron donors are organic carbon and or hydrogen. This is trivial knowledge. What is important is about external electron acceptors. That could be sulfate, thisulfate, sulfur, nitrate, etc. Why they are important? Because sulfate reducing bacteria, they reduce sulfate to sulfide. They don't eat sulfate. By doing these chemical reactions, they use the electrons for themselves. And if they cannot find sulfate, but in your system, as a corrosion inhibitor, you have applied nitrate, they will use nitrate. So an engineer comes to me and says, Reza, I'm sure that in our system we don't have SRB because we did a chemical analysis of the system, we don't have sulfates. You know the answer. And main difference between fermentation and this anaerobic respiration is that in anaerobic respiration, the electron acceptors are external. In fermentation, they are internal. But this is an issue for microbiologists, not us. Then temperature. If bacteria can survive the can survive within room temperature, they are called as mesophils. If they survive at higher temperatures, they are called thermophils or so-called archaea. In Australia, I have seen bacteria isolated from reservoirs at a temperature of around 120 degrees centigrade. So high temperature is not necessarily an obstacle to bacteria. And then we have psychrophils that prefer low temperatures. And one thing, let me tell you, which is not related to microbial corrosion, in your houses, the dirtiest place from a bacterial point of view is your refrigerators. Some examples of CRB, CRA. What are these CRB, CRA? When you read materials by self-acclaimed MIC experts, they try to give you the impression that all these bacteria that we know from MIC literature, they are there to enhance corrosion. No, not necessarily. Sometimes they decelerate, they slow down corrosion or even inhibit it. That's why I invented the term CRB, corrosion-related bacteria or corrosion-related archaea. They are related to corrosion, either by enhancing it or the other way around. Examples are SRB, sulfur-reducing bacteria, sulfur oxidizing bacteria, iron oxidizing bacteria, iron oxidizing, and Clostridia, but this is not all. These bacteria and archaea counterparts, they have been isolated from waters in the Persian Gulf. And currently we know that there are at least 50 types of microorganisms that could be related to corrosion. For more information, please read my book, Biological Treatment of Microbial Corrosion. It is also interesting that when it comes to microbial corrosion treatment, most of the time people's mind go to using chemicals, biocides. However, in this book, we have described that it is quite possible that you use bacteria against bacteria without involving chemicals. So when you see that there are a lot of bacteria involved, then you see, especially on the internet, that someone says that SRB are the main bacterial concern. No, they are not. They are not, my friend. You are wrong. And what is biofouling? In Australia, Scotland, and many other parts of the world, I have seen these so-called orange bloom. They normally happen in shallow waters, and we still don't know what they are. However, we do know that sulfur-reducing bacteria are also involved. What happens is that behind this growth, very severe pitting happens, and mechanical integrity of steel piles are actually gone. Biofouling, what happens in biofouling is very simple, although it is a pain in the neck for many industries. First, a biofilm is formed. This biofilm acts like a glue on which other objects, including marine organisms such as barnacles, etc., are actually stuck. So if in any way you could actually prevent biofilm formation, then chances are that biofouling will also be adverse. Biofilm formation, this is all the story. Everyone knows that, yes, we have planktonic bacteria, slide number 2. Planktonic means freely swimming. These bacteria, they, as long as they are in the bulk water and can find food, they stay in their planktonic state. When the food actually sinks or falls down on surfaces under the impact of its weight, the bacteria realize this via a sense it is called as quorum senses. They realize that there are other bacteria around. They realize that food is not in the vicinity, in the reach. They have to go down and find it from the surface. Then bacteria come and actually start to sit on surface and become motionless. They are called sessile bacteria, plate number 4. And by time they become dead, because the biofilm starts to increase in thickness, and therefore ingress of oxygen and food to the bacteria living at the interface of biofilm and the metal surface will become restricted, and the bacteria will die off. But okay, they die off. However, their bodies are still there, like zombies. So any change in the hydrodynamics of the fluid will cause them to be taken off. So you will create areas of high and low differential oxygen pressures that it is called differential aeration cells, where high oxygen pressures will create anodes and low oxygen pressures will create cathodes. The thing is that here, one important issue is that, for example, in a pipeline, for example, a fire water ring, you have developed biofilms, for example, because water stays there stagnant for a long time. And then somebody comes and says, oh, yeah, I have read somewhere that keep the water flowing. He operates the pipe, the pump, and then water starts to move. A part of the biofilm is taken off and travels to the other part of the ring, which by any chance has been left cleaner. And then that part also becomes contaminated. Okay, for those of you who have listened to me carefully so far, what mistake I deliberately made here while I was explaining the mechanism? To give you a clue, I made this mistake when I was talking about anode and cathode formed because of differential oxygen pressures.
You said something about high oxygen pressures was the anode and low oxygen pressures was the cathodes. The normal thing is the other way around.
Exactly, thank you. It is exactly. When the oxygen pressure is low, that spot becomes anode. And that's why peating normally happens under the biofilm, not outside of it. Thank you. These are two examples of real biofilms that I have grown on carbon steel by SRB and iron-reducing bacteria. You can find more about it in my book, second edition of my book, Microbiologically Influenced Corrosion and Engineering Insight, published by Springer. One thing is interesting that I will use in my next slide. As you can see, there is nothing whatsoever that would resemble these structures as films. They are not films. They have a patchy structure. So in 1978, the term biofilm was suggested. The term brings to mind that it is totally biological and it is a film. Neither is true. Only a very small percentage of so-called biofilm is biological. The rest is non-biological material. That's why if the TDS, total dissolved solid, of your fluid is high, then the likelihood of getting biofilms will be high. And it is not a film either. That's why in 2020, I published a paper in which I proved my point, why biofilm is a wrong term, and instead I suggested the Greek name temenos. Temenos in Greek means cut off. And the reason I invented this term is that when the biofilm is formed, under biofilm conditions could be vastly different from conditions outside the biofilm. Outside the biofilm, pH could be 7. Within the biofilm, it could be acidic. Outside the biofilm, it could be oxygenated. Under the biofilm, it could be anaerobic. So from now on, instead of the wrong term biofilm, I will use temenos. MIC treatment, one important thing, my friends, is this. Main point in microbial corrosion management is having a strategy, not the tactics alone. What do I mean? Screening and understanding mechanisms are a strategy. What is screening? Simply, it means that you have to learn your enemy. What type of bacteria you have in your system? Are they archaea mainly? Are they salt tolerant? Are they only sulfur reducers? What are they? Screening. Then you have to know the mechanisms. As we will see in a moment, even for sulfur reducing bacteria, the mechanisms could be quite confusing. So know the mechanisms. Then go for tactics. What type of treatment I have to use? What type of monitoring I have to use? Industry, just because they don't care about training and research, they don't care about the strategy. I am telling you because of my 30 years in industry worldwide, in many industries, from oil and gas to power generation. When they come to me, they say, Reza, we have this severe pitting. What we can do about it? Can we do coating? Can we do biocide application? They even don't care what the root cause of the problem is. So, my friends, you do not make the same mistake. First, learn the strategy, then go for tactics. There are basically 8 methods by which both MIC and non-MIC corrosion can be treated. Chemical, for example, inhibitors for non-MIC and biocides for MIC. Physical, like coating, mechanical, pigging, electrical and material selection, design, modeling, training. I will just go through CP, which is our topic today. Candidate protection is a very tricky issue, as you all know. This is an example from a case we had in Australia for the same topic based on 4 approaches, 4 standards. We got 4 different budgets. So it is important to know which version of which standard you are using. I'm not a CP specialist, but I do suggest that at least use 3 standards and compare. Then, is CP always effective against MIC? Do you want my honest answer? I don't know. And anyone who says I know, I challenge him. NACE standard in 2006 said that you can go for a minus 100 millivolt, what you call it, polarization to be safe against MIC by SR. Then in 2013, SP0169 said, no, if it is MIC, you are allowed to go further down, minus 300. And God knows, in 2033, what they would say. Then when it comes to offshore industry, my friends, I could gather 9 standards from ISO to BS. And all of these have their pros and cons. Because this presentation is recorded, I'm not going through all of this. You can read these for yourselves. However, there are the 3 important points that is common for all of them. Out of a range of various potentially corrosion microbes, only SRB are addressed. Those who have written these standards seems have not been aware of anything other than SRB. Some of these standards are silent and do not consider MIC. And I'm sorry to say that DNV RP B401 edition 2021. I'm not talking edition even 2000, edition 1900, 2021. Can even be considered as handicapped. I'm sorry for being so direct, but DNV standard is one of the standards I do respect, but unfortunately. Mostly the protection criteria is minus 0.9 volts against AgCl seawater. And this minus 100 millivolt potential shift comes from a paper in 1925. Can you imagine? It is absolutely absurd. You see the evidence here, and we do need to do something. Why cathodic protection could be effective against MIC? This is my theory that I have explained. You may say that, Reza, it is rubbish. At least I tried. What I said was very simple. There are two issues related to cathodic protection that could affect cathodic protection. One is electrostatic. Bacteria can be regarded as negatively charged particles. When you apply cathodic protection, you actually energize the surface by negative charges. So there is a repulsion forces, all right? That's why cathodic protection could be effective against MIC. However, according to a concept we have in material science, when two opposite charges come too close to each other, around 0.4 nanometer, repulsion forces becomes attractive. So maybe this is why sometimes cathodic protection works against MIC and sometimes it does not. Then we have the fact that when you have CP in place, hydroxyl ions are produced, so the pH goes up, and then, of course, bacteria become, actually, it is against the way bacteria can work. I'm not going through using higher mathematics to model microbial corrosion. I'm just showing it to you. Unfortunately, mathematics have not been used extensively. Training anywhere when they want to cut the budget, what they do, we don't need training. We are absolute. We know everything. And it is interesting if you know that every 20 days worldwide, 16 new papers on MIC are being published. MIC tips: any system with a stagnant, untreated water that comes into contact with a relatively rough surface could be vulnerable to MIC. If you have a case of corrosion and you can't explain, don't jump on the, don't involve bacteria right from the beginning. Try to explain it by non-MIC mechanisms. If failed, then go for MIC. Don't do it. And the conclusion is this: a strategy first, then tactics. Then, contrary to its name, MIC is not to be dealt with corrosion knowledge and experience only. Focusing too much on non-chemical features can mislead you. Running after new tactics could mislead you. Don't go for that. Don't go for the fashion. Don't obey the trends. They say that, oh, nowadays we have to go for nano composites. So let's go for nano composites. This is absolutely absurd. Most important of all, if you think it is expensive to hire a professional, wait till you hire an amateur. The challenge: there are two buried pipelines, one in clay, another in sand. The first question: in which one you accept microbial corrosion? In both. For which one SRB? In clay. Why? Because not because of moisture, because it has less oxygen. Thank you very much.